So I'm really happy to be here and kicking off the first webinar of 2023. I hope everybody is having a safe and productive winter and that the days are starting to get longer in your neighborhood and you're getting outside to enjoy everything the season brings. Like Christy said, I'm talking about this project that we've been working on for the last few years now, networking the spatial structure of Boreal Caribou populations. And before going any farther, I'd just like to acknowledge that this has been a big project with a lot of contributors. It was recently published in Global Ecology and Conservation. Christy's going to drop this DOI link into the chat so you can access the original paper if you have an interest in following up on exactly what we were doing. I've listed our co-authors there and also the data contributors because this was a big collaborative project because we're covering a large area. And the research was funded primarily through FRIA as well. So very simply, what was the objective of this project? And very simply, it was to develop a data-driven approach to better define Boreal caribou ranges in Western Canada. And so why do we want to do that? Well, as anyone who has looked at the ranges will realize that there have been some arbitrary decisions in how we've set some of these boundaries. Many of them come to abrupt halts when we get to the boundaries with Saskatchewan and Northwest Territories and BC, obviously. But in other cases, we have these transboundary herds like we do in the Chinchaga, for instance. And so in some cases, there's been some decisions that don't necessarily make a lot of ecological sense. And so I think it's always worth revisiting some of the way we have arranged these things. And probably more important is the fact because we're doing recovery planning within the bounds of these areas, there's important implications in terms of assumptions we make around geographic closure. So what that means, of course, is if we have a range and we're doing recovery planning in that range, we're calculating demographic risks to that subpopulation based on those boundaries. But of course, if the caribou within that range, that subpopulation LPU that we have defined, if the caribou are actually more connected than suggested by that range boundary, then there's a chance that maybe the demographic risk is lower than we're assuming based on that boundary. At the same time, if they're actually more fragmented than suggested by those LPU boundaries, then perhaps the demographic risk is higher. So what we wanted to do was try to figure out whether or not we could do a better job with the data we have in order to define these ranges. So obviously there's significant data available. We've been collaring caribou for 40 years now, which is quite amazing. And certainly over the last 25 years, the majority of those, the data collection has been with GPS collars. So we've been collecting a pile of information across a bunch of jurisdictions. So the question is, how can we best harness all those data that we have collected to do a better job of trying to define the spatial distribution of caribou through these areas? And it's also important to understand that even though we have these boundaries, we know that caribou through much of this are still in a lot of areas continuously distributed. So, there's places of higher concentration and lower concentration, but they are certainly still in a place where they're moving somewhat continuously through large areas, and so there's a real challenge in trying to understand how we might logically break up some of those ranges into logical areas for management. So that was the general approach. Now, of course, we're not the first people who have thought about this. We're standing on the shoulders of giants when it comes to this work. And obviously, when we think about this, the first thing we think about is, okay, well, we need to find some way of clustering all this telemetry in space and time in some way to identify logical groupings of caribou. And we weren't the first to think about that. People have done it with other species. Maybe one of the first applications was for polar bears in the Canadian North, but a little later, Jen Shooter and Art Rogers did this paper for the Winnipeg North American Caribou Workshop in 2012, where they basically did this kind of approach using a hierarchical clustering method for caribou in northwestern Ontario. And in this case, in this application, the idea is to look at the median X location and the median Y location within each caribou for each year and each season. And so what happens is basically you take the full distribution of caribou movements and you thunk them into a single point in the X and in the Y across different caribou seasons. And so we started with that. And so this is an example of the data table that we ended up with. And I'm putting this up for a specific reason that will become clear, but this is the idea that in each caribou year, this is the names of our caribou here by an identifier in a year, each row in our data frame is an observation, and each column here, calving X, Y, fall X, Y. So each season and an X and a Y location, those are the random variables. And that's the basis for this hierarchical clustering. So we did this analysis, and it works well, although you lose a lot of resolution in the data. Obviously, with GPS telemetry locations, we're collecting a lot of high resolution data, but you lose that if you're just using the median X and Y location. So we were looking at some way of making a better use of all the data that are available in order to to bring some more resolution to the analysis. Now, this is a good approach, most obviously because it works, because you can actually do that clustering. The problem is if you start getting to finer resolution location data and you start adding, say, hundreds of caribou into the analysis, you get into a factorial problem and your data matrix gets way too big very quickly to analyze. And so even though we've had 10 years of advances in computing power since this paper came out, we're still nowhere near close enough to be able to do this kind of approach using, say, every location on an animal rather than using these medians. So we had to come up with some sort of different way of doing the analysis that would take into account the full range of where caribou are, use all those data to the best extent possible, but not run out of memory on all our computers. And so the approach we did is we essentially kind of flipped the data matrix. And so I'll take a moment to explain to you what that means. And so if you imagine, and what we did essentially is we started by gridding the landscape into cells and considering each cell on the landscape to be an observation. So very simply, if we just think of a landscape up here in the top left, where we just have 4 grid cells, and we have 2 caribou, we got a red caribou and a blue caribou. And in this example, all the red caribou locations are in one cell, and we have the blue caribou spread between these 3 cells. Now, the only difference between this top representation and the bottom representation is that I've moved one of the blue dots from the lower left to the top left. And so if you think about this kind of conceptually, in the bottom example, the red and blue caribou are slightly more associated in space than they are in the top example. So we have this kind of conceptually is the way we're going with this. If you have caribou locations, if you have different caribou that are more associated in terms of the grid cells they're falling into, then that suggests that they are more related in space. And so this is what we were trying to get after. And we can basically come up with a data matrix where each row in our matrix is a grid cell. And then we just simply count the number of observations from each of the caribou that are falling into those grid cells. And we end up with each caribou is actually a random location, a random variable. So completely different from the previous data matrix where we have observations that are caribou years. Now caribou are actually random variables. And rather than the random variables being XY locations for caribou. So that's basically how we set it up. I won't go into the math around this, but basically what we're trying to figure out is how closely associated are those columns for each one of those caribou. And information theory, going back to Claude Shannon in the late 1940s, came up with a lot of the math that can be used to calculate how closely these animals are related in this kind of non-parametric way. Now, I mentioned here that we use 10 square kilometer grid cells. And so that's kind of the Goldilocks zone we found. We did try different grid cell sizes. So we went down to 5 square kilometer grid cells And we also tried 25 square kilometer grid cells. We found when we went down to 5 square kilometers, we basically got the same answers, very similar, but we had 4 times the data to analyze because, for instance, if these were 10 square kilometers here, you can see if we had 5 square kilometers, we go from 4 cells to 16 cells. So 4 times the cells we have to analyze, but no real change in the answers that we were getting. So we dropped that just to conserve computer time and our sanity. At the same time, if we went up to a broader grid cell of 25 square kilometers, you can see with this example here, both those caribou would be entirely located in one grid cell, and there'd be no differentiation between them at all. And that's what we found when we went a little higher. We started to lose spatial resolution. So it's not particularly scientific in an objective measure. I mean, I guess we could have tested a lot of other grid cell sizes in between there, but it was kind of where we lined up. And in terms of the scale at which caribou move around, that seemed to be the right scale. Okay, let's talk about the data set. So we did basically what we could to get a hold of all the GPS telemetry data that we could possibly come up with, not just from the province of Alberta, but also from the adjacent jurisdictions. Lots of collaring in Northwest Territories, lots of collaring in the Boreal Zone in Alberta, and of course, Phil McLaughlin's lab's work from Saskatchewan, from northern Saskatchewan, the SK1. We acquired all those data from him as well to do this, to integrate into this analysis. And there's different relocation frequencies on some of these animals. So, you know, some people, in some groups would program the callers to collect, say, 5 locations a day during calving and then drop down at other times a year. Other callers were once a day, other callers were twice a day. So to deal with that, we only looked at the first location on each caribou for each day. So our analysis is actually based on telemetry days. And for that, we had 891,000 telemetry days collected on nearly 1,600 caribou from 1998 to 2020. That was our data window. Obviously, there's piles of caribou still collared out there, still acquiring information. So if we were to redo this now, we'd have, again, a significantly larger data set, chances are. One thing that we didn't do is we didn't stratify by time, because you might be thinking, well, 1998 was a long time ago. There were a lot more caribou around there, and that's true. When we did look at various time slices of these data, the distributions didn't all change all that much. So even though there were more, except in a couple of spots, in general, data from the late 1990s, early 2000s, the telemetry is still showing up in the same places. So there isn't really kind of a lot of evidence of super recent range contraction. There's just fewer caribou there in the same places. So that's kind of what we're seeing. So we just pooled all of that stuff together and did it as one big analysis. Now, in terms of the actual techniques that we used, we stole shamelessly from other fields of research. And maybe not surprisingly, this is an active area of research, the idea of how you take kind of multivariate networks or multivariate data and see how they are networked together. And not surprisingly, there's a lot of work being done on this for humans, which is how do you build connections between people based on a pile of data that you have? And where are people getting those piles of data? Not from telemetry, but from Facebook and Google and Instagram and TikTok. And all of these companies that are stealing all your data all the time, probably right now while you're on the phone or have a browser window open, you know who you are. But all those companies are taking data from you all the time, and they're thunking that into connecting you with other people and building these networks. So there's a lot of innovation that's happening in this, but there's no reason why we can't apply those same techniques to telemetry information. So very specifically, what we did is it's called an undirected, because there's no arrows on the lines between these dots, but undirected one-to-many Gaussian Bayesian network. And all that means is that each caribou, which is represented by a node in the network, can be connected to more than one friend. So this caribou here is connected to 7 other friends who then are connected to other friends, etc. And And so that's the basic approach. And then there's a learning algorithm. And the learning algorithm basically tries different configurations of edges. We call the lines between the caribou, those are edges. And the network just basically tries all kinds of different configurations of edges and tries to, and much in the same way with information theoretic methods that you may be familiar with if you've worked with AIC when you're doing resource selection functions and that sort of thing, is that it's trying to optimize the network to capture as much variance as possible in the network while using the minimum number of edges. So it's imposing a penalty on a network score every time it adds an edge, but every time it adds an edge, it's explaining more variance. So it's trying to optimize those two things to come up with the optimum network. And what happens is that not all caribou are networked together, because some caribou are too far apart from each other, or in some cases, there's not enough locations where there's not enough information to indicate that they're connected to other caribou. And so then we end up with these somewhat isolated subgraphs. So in our case, when we started with these 1,500 caribou across this big range, we ended up with these 24 large subgraphs that had more than 5 caribou in them. If they were less than 5, we didn't really consider it to be any kind of meaningful group. And a lot of those subgraphs of less than 5 tended to show up on the edges of the range, although not exclusively. And so there's a bit of a slop in there, but we were mostly interested in what the big picture was telling us. And for those of you who are analysts and are interested in doing this kind of analysis, we use the R packages BNLearn and iGraph. BNLearn for this particular network analysis, and iGraph for the graphing, and also for the next level of analysis that I'll show you in a moment. So pretty accessible, obviously freely available in the paper that's posted in the chat. I provided all the code for that. Okay, so what the results look like. Well, what I've done here is I've colored up all the different subgraphs as, all the telemetry in different subgraphs are a different color, and then I put MCP home ranges, or 100% MCP home ranges around the 24 largest subgraphs. And so there's a few things that come screaming out, obviously, is this big giant blob we have here that covers not only NWT, but also comes right down through most of the ranges in British Columbia, even stretching down into the Chinchaga, and also continuously in Alberta through Bishtu and Yates and Caribou Mountains. And so we think it's coloring up this way because this is when I was talking about caribou basically being largely continuously distributed throughout this whole area. At this level of analysis, there's very little to distinguish them as different groups. And we have the same phenomenon happening in Alberta to some extent, down through Red Earth, Westside Athabasca, even crossing the Athabasca River and incorporating some of the Eastside Athabasca as well. At the same time, we do have some things that are coming out distinctive. So the little Parker Range in BC comes out as separate. The Chinchaga, for the most part, is also showing up. Richardson is showing up distinctly, but is obviously, unsurprisingly, transborder with Saskatchewan. Things get messy along the southern edge of Caribou Range in Alberta, and we think that this is an indication of just the ongoing fragmentation and extirpation of caribou along that southern boundary. I don't think anybody would be too surprised to see that kind of thing, so it gets messy there. We also found some structuring in Saskatchewan, but the caveat here is that's only based on a few years of data from that SK1 study, so just about 3 years of telemetry. So whether this would be stable over the longer term had more telemetry be collected, it's hard to say. So I think that's kind of a tentative structuring that we're doing there. So kind of interesting results at this level of analysis, but we wanted to dive a little deeper to see if we could resolve some finer level structuring, because as interesting it is to have nearly 800 caribou grouping up into this large range that runs right from, this huge area right from Chinchaga up to north of the Mackenzie. the question is there evidence for finer scale structuring? And to do that, we did a second level of analysis, and this is a level analysis called community detection. Again, borrowed shamelessly from those who are analyzing human networks, lots of literature around this. And what the community detection analysis does is it starts with a subgraph, and then it looks at the density of arcs within that subgraph. So you can see in this particular subgraph, there's places in this subgraph where the caribou are much more connected together than in other parts of that subgraph. So based on a measure of what's called modularity, that's the technical term for it. But again, it's just kind of looking for an optimal solution in terms of how much variance is explained in the graph. And this again applies another algorithm on the existing graph. It's not growing the graph by adding links, but it's analyzing the links that are existing. And the one we use is it's called the fast greedy algorithm, and it's a clustering algorithm, not dissimilar to the algorithms that were used in the earlier analysis, for instance, that Jen Shooter and Art Rogers used. And so what we did is we used this to see if we could look for finer scale structuring within these large subgraphs where we had more than 50 caribou to see if something made sense. And so I'll show you the results of that in a couple of slides. So here's that big blob that we had in the northwest of the range, Northwest Territories is extending down. And you can see now, even though I've put the MCPs of the big groups on there, now I'm showing the community detection within that, and we're starting to see a lot more structuring. And most interestingly, we start to see some significant breaks along major terrain features, so major rivers. So for instance, in the Mackenzie River is basically serving as a break between different communities. Also in Northwest Territories is the highway. It's not illustrated here, but it's running the highway Highway 3 up to Yellowknife runs between these two communities that were detected as well. Also, Hay River is serving as a bit of a barrier between different groups, and the Petito in BC, and the Fontas too, as well, farther south in BC. So starting to show up some indications that starting to show some breaks along landscape features that we suggest are important for distinguishing different groups of caribou. At the same time, where there aren't those kinds of features, we are seeing the way that caribou are structuring, say, across these jurisdictional boundaries. So most interesting, of course, is I have the original ranges underneath here, but obviously the Calendar and the Bishtu and the Southern Jaycho from Northwest Territories, all kind of separate in terms of our LPU range planning. But this analysis is showing in this blue blob that actually the west side of the Bishtu, all of what we call the Calendar range in BC, plus the Southern Jaycho, are all grouping up into one community. Not surprisingly, that caribou are moving across the boundary and doing that. Now, in other areas, we do have ranges that are also remaining fairly distinct. So the Max Hamish in BC, largely the way we map that, the Snake to some extent, although we're showing a separate group kind of between those, Caribou Mountains as well, but also some movement between Yates and Caribou Mountains, and that Yates group is actually transboundary with NT and running straight up on the east side of the Hay River. Kind of a lot of more interesting structure that's happening here. Also the boot down in the Chinchaga, and we have good evidence that group is pretty isolated. If there's even any caribou left in there, I'm not sure. But yeah, so kind of interesting the way this is. Oh, one more thing, I guess, is also this is the profit range here, and so we can see that there's quite a bit of telemetry that's been collected right outside of the current range boundaries of caribou as we're defining the federal recovery strategy. So we have this group of caribou that are ranging from the profit right across kind of what we would have thought as no person's land, I guess, between caribou ranges, and right into the northern part of the Chinchaga, we're seeing a continuous distribution of caribou along there. So there's some places here where I think it becomes obvious where there should be some boundary changes. Move over to kind of the core of the Alberta herds and we can see again a lot more structuring, and it gets even more confused along this southern boundary. Gets pretty messy in here, but we do see some better distinction between red earth and west side, and then the east side Athabasca is actually showing up as a different group here. So we are getting some structuring, some substructuring here. There's people who are far more familiar with the data than me in this particular area and can probably point to places where this makes sense and maybe places where it doesn't make sense, but it is certainly messier and a lot less clear here than in the Northwest, but that could reflect the relative condition of caribou in both those areas as well. Okay, so that's the main pieces that I wanted to talk about today. In terms of implications of this work, so I think it's clear that that some of the existing LPUs that we have are lining up with this analysis, but not necessarily at a consistent network or community scale. So in some cases, we have the big scale analysis and a range like the Parker is coming out and the Chinchaga is kind of coming out and saying, okay, well, that's aligning with this range, this kind of network scale analysis. At the same time, we have to get down to that kind of community detection analysis to start seeing the differentiation between, say, Yates and Caribou Mountains or red earth and westside Athabasca. So it seems like the way we have defined these LPUs kind of straddles those two levels of analysis. But I think one of the benefits, obviously, is that we have been able to do a better job across these jurisdictional boundaries that have traditionally broken up our ranges and shown how those animals are moving back and forth. So I think we have provided some evidence that at least in the Northwest, we have caribou that are likely more connected than is suggested by these LPU boundaries. And because they're more connected, we're clearly violating this concept of geographic closure, and as a result, those caribou populations may have slightly lower risk. But of course, in other places, we're seeing this very complex spatial structuring, which suggests that they may be at higher risk. And so, again, if we look at a herd like the Chinchaga, the isolation of those caribou down and kind of the boot of the Chinchaga probably suggests that that particular subgroup within the Chinchaga is much higher risk than, say, the Chinchaga range as a whole, which is the scale at which we're doing the analysis. So I think that this is useful. I think the other thing to point out is that we haven't done a deep dive on this, but there's obviously opportunities to tie this in with other sources of evidence. So we've had some discussions a little bit with, say, the Delineating and their knowledge of how caribou move among Bishtu, Yates, and Caribou Mountains, and it's kind of lining up with their knowledge of how they think caribou move rather than in these discrete ranges. Also, there's ongoing genetic work, and the genetics work is in a lot of ways similar to this, looking at networks of connectedness amongst individuals based on genetic relationships. Had some initial discussions talking about how some of these results are also, that there is some concordance between what we're showing on the spatial side and what's showing on the genetic side. So I think there's opportunities in the future to bring these three sources of evidence together and make a stronger case for how we should be structuring our LPUs in the future. Kind of a long-term project, but I think it points to there's certainly lots of opportunity for some improvement. Benefits and limitations, as always, in these kinds of analyses. So first, on the benefits side, well, I think whenever we're using data, we certainly have an opportunity to be more defensible in how we're talking about spatial structuring, particularly if we can correlate it with these other sources of evidence, be it traditional knowledge or genetic evidence. And I think this has also demonstrated how we can determine some spatial structuring even within populations that seem to be fairly continuous. So say that group that is Callender, west side of Bishtu, and southern Decho, that's a group that shows up, but within a more continuous distribution that also extends out to the Max Hamish in the west.
Yates Caribou Mountains in the east, and the North Facho as well. So I think we can start to see how these groups, that there is some evidence of spatial structuring even within these continuous distributions. But obviously the analysis also has some limitations. So first of all, there's going to be sensitivity to gaps in the sampling. So if we don't have any telemetry in a place where there's caribou, obviously that's going to show up as a whole, and that's going to affect how the distribution is being reflected. And so it's really important to have good sampling everywhere. And of course, we never have great sampling everywhere. So we always have to take that into consideration when we're doing this kind of analysis. There can be some sensitivity to outliers. I clean the data as best as I can, but if there's telemetry errors or say long distance movements by individuals, that can affect results somewhat, although it's fairly robust to this because this is an information theoretic approach, right? So it's not just one distant location is going to throw out the analysis. That's probably going to be ignored. And I say probably, which leads into the third piece, is that this is a computer doing this through a learning algorithm. And so the results are not always transparent. People say, well, why did it group up in this particular area in this particular way? And often I don't have an answer for that, because who knows how some of these things are being optimized. So there's a bit of a transparency issue there, which also leads to the problems around some confirmation bias. We tend to look at the distribution and say, well, this follows our expectations, so we think that's true. This area, we're not sure what's going on there. We're just going to kind of ignore those results or kind of hand wave it away. We've got to be careful of that sort of thing when we're dealing with this sort of analysis. So there's some judgment involved. But overall, I think that we've moved the needle on our understanding of how subpopulations are structured in Western Canada. And I think that there's some clear next steps for how we can integrate it with other information to do a better job of representing what these ranges look like. So that's all I have for you today. Thanks very much for dialing in, and I'm happy to take any questions.